Hello, students. Okay, we're going to go ahead and finish up the last set of notes in Unit 3, the 3.3 notes on photosynthesis. In this video, we're going to look at Topic 3, which is about the Calvin cycle. So in Topic 2, we talked about the light reactions, or the light-dependent reactions, which take place in the thylakoid. And in those reactions, we ended up making a few different things. We ended up making some NADPH, who's carrying these high energy electrons that were energized by these photosystems and ultimately came from water. We did make some oxygen, which is the result of water losing those electrons at photosystem 2, and that oxygen was produced. And we also were able to make some ATP during the light-dependent reactions. And so the NADPH and the ATP, those are going to move on to the Calvin cycle. We're going to see them now in Topic 3, why those were needed are made here in the light reactions. And then the oxygen, that's now just released to the atmosphere, so we're not really going to see him again. So in the Calvin cycle, let's go back to that, there's going to be a series of chemical reactions that take place that start and end with the same molecule. And so the starting molecule in the Calvin cycle is this 5-carbon molecule called rubip, or ribulose bisphosphate. And what's going to happen in this cycle is we're going to start building some sugar molecules by putting together smaller molecules and using ATP and those high energy electrons from NADPH. We're going to need the ATP and those electrons from NADPH to start putting together smaller molecules to start building sugar. And so in this cycle, what we're going to see is that we're going to see an input of small molecules in the form of CO2, carbon dioxide. And then as a result of this cycle occurring, we're going to see an output of a small sugar called glyceraldehyde 3-phosphate, or G3P, is going to be produced in this cycle. And in order to make one G3P molecule, which is a 3-carbon molecule, the Calvin cycle is going to have to happen 3 times. So every 3 cycles of the Calvin cycle, you're going to end up making one G3P. So we're going to go ahead and go through what happens here in the Calvin cycle. It can be broken down into 3 phases, and you guys just need to know the gist of what's going on in each of these phases. So we'll start with the carbon fixation phase. This is the first phase of the Calvin cycle. And what happens here is this is where rubip, this 5-carbon molecule, is going to be joined with a carbon dioxide molecule to form this 6-carbon molecule. And then this 6-carbon molecule, and then the next reaction will break in half into these 2 molecules here called 3-PGA. And when we go through this in this diagram here, we're going to go through this cycle happening 3 times at once. So everything I just said, we're going to pretend that it's happening 3 times at the same time. And so we're taking not just one rubip and one CO2 to make 2 of these guys, but we're going to actually take 3 of these guys and join them with 3 CO2 molecules to make ultimately 6 of these guys. So that's why you guys see these numbers here, because we're going to go through what would happen if the cycle is happening 3 times. And you'll kind of see why we have to talk about it happening 3 times at once soon. But that's called the carbon fixation phase. So putting together these rubips with CO2, forming these 3-PGA molecules right here. This is accomplished by an enzyme called rubisco. So rubisco is the name of the enzyme who catalyzes the reaction that joins CO2 with rubip. And this is a really important process here that happens in biology, where this is where plants are going to get the carbon from the air. There's carbon atoms in the air in the form of CO2, and now this carbon is going to become part of the plant and the plant's organic molecules inside of the plant. And so now officially after the carbon fixation phase, that CO2, that carbon that existed in CO2 is now carbon that is part of the plant, and it's part of the organic molecules that are inside of the plant. So we call that carbon fixation because that carbon used to be free in the atmosphere flying around in these CO2 molecules. But now that carbon is in a fixed position and now is part of this cell and this organic molecule of this plant. Like, this is where it is now. And this is super important because this is where plants get all of their carbon. So all life on this planet, including plants and us and bacteria and all cells, we are carbon-based life forms. All of our cells and all life on this planet is made of mostly carbon. in plants, they get all of their carbon, 100% of their carbon comes from the air. Like this is where they get all of their carbon that's going to be used to build all their carbohydrates and proteins and nucleic acids and lipids, all the organic molecules that make up the plant and the plant's body tissues and the plant cells, like the whole structure of the plant. All of that carbon, they're getting right here from the air, from these CO2 molecules in this process called carbon fixation, which is then going to now put that carbon into these organic molecules that the plant is then going to continue using and changing in lots of different chemical reactions to make everything that the plant needs to build itself. So anyway, that's the carbon fixation phase. And then in the next phase, we have what's called the reduction phase. In the reduction phase, we're going to take those 3 PGA molecules. This is a 3 PGA molecule, and this is a 3 PGA molecule. And we're going to reduce those molecules. Which is why we call it the reduction phase. Reduce means gain electrons. So these 3 PGA molecules are going to gain electrons, and they're going to become these molecules here called G3P. And so when you look at these guys, they look like they're the same thing. This is a 3-carbon molecule with a phosphate, and this is a 3-carbon molecule with a phosphate group. They are similar molecules, but they're not exactly the same molecule. So just know that I'm not showing you the whole chemical structure of this molecule and that molecule. They are different molecules, even though they look the same in this diagram here. But basically, again, in the reduction phase, we're going to take these 3 PGA molecules, and they're going to gain electrons and become G3P. In order for that to happen, there's a couple of reactions that take place. Those electrons are going to come from NADPH. So NADPH, which was made in the light-dependent reactions, is going to take those and has those high-energy electrons. We are going to now give those high energy electrons and drop them off here so that they can join with these molecules to become G3P. And then that NADPH, when it drops off those electrons, becomes NADP plus. So that's the reduction part happening here, where this is now gaining those electrons as this guy drops off and loses those electrons. And then this also requires some ATP, as you guys can see here. So again, some of that ATP that we made in the light reactions, we are now using here in the Calvin cycle during the reduction phase. And on here, you guys will see these numbers here, because again, we're looking at as if this cycle was happening 3 times at once. So in the first phase, we had 3 molecules of rubip joining with 3 molecules of CO2, becoming 6 of these molecules of 3 PGA. And then in the reduction phase, all 6 of those molecules of 3 PGA will become 6 molecules of G3P, 5 of them, and then there's 1 right here. And the reason why we are talking about this happening in sets of 3 is because every 3 Calvin cycles will end up producing 6 G3Ps, like I just showed you. So when this cycle happens 3 times, we're going to have 6 G3Ps. And the rule is every time you have 6 G3Ps, one of those G3Ps gets to leave the cycle. So once you have 6 G3Ps, which will happen when you've done the cycle 3 times, you now will have a leftover G3P that gets to leave the cycle, and then the plant cell gets to use this G3P to build lots of different things, including glucose, sugar, and other organic molecules. But the other 5 G3Ps that we made, they have to continue on into the last phase of the cycle, and they're going to be turned back into rubip. So in the last phase, we have what's called the regeneration of rubip phase. And in that phase, we're going to take those 5 remaining molecules of G3P, and we're going to turn them back into the 3 molecules of rubip that we started the cycle with. And so if you count the carbons here, there's 3 carbons in G3P. And if you have 5 of them, that would be 15 carbon atoms in all 5 of these molecules. And if you look at rubip, the 3 molecules of rubip that we started to do the cycle 3 times, those 3 molecules of rubips, they each have 5 carbon atoms. And if there's 3 of them, that would be 15 carbon atoms. So basically what's happening here in the last phase here, the regeneration of rubip phase, is we're taking those 5 molecules of G3P and we're just rearranging those molecules into these 3 molecules of rubip. We're taking all these carbons and rearranging them into these guys that we started the cycle with. And to do that, it does require some ATP, a little bit more ATP to finish this off in these last couple of reactions here in the regeneration of rubip phase. And then you officially now have the 3 rubips that you started with. So then the cycle can happen another 3 times with another set of 3 CO2 molecules that enter the cycle. And then you can make another set of 6 G3Ps. One of them gets to leave, the other 5 go back to being 3 rubips. And then you can do the cycle another 3 times, and another 3 times, and another 3 times. And every time you're doing the cycle 3 times, it's going to require 3 CO2s that go into the cycle, 1 per cycle. And then when it happens 3 times, out comes a G3P molecule. So in a somewhat complicated way, the cycle is kind of accomplishing something simple. It's basically just taking these 3 carbon dioxide molecules, which each contain a carbon. And in the cycle, those carbon dioxide molecules are being put together to form a molecule of G3P, which is a 3 carbon molecule. So 3 carbon dioxides go into the cycle and 1 G3P molecule comes out of this cycle. So that's the overall gist of what we're accomplishing here in the Calvin cycle. And so then this G3P, like I said, the one that's left over, the one that you make every 3 cycles, this 1 G3P that gets to leave, this is now going to go to the plant cell, who's then going to use this G3P to build other organic molecules. Most commonly, it's going to use this to build glucose. And so real quick before I wrap this up here, though, I just want to point out a couple more things. This whole series of reactions, the Calvin cycle, you guys, this takes place in the stroma of the chloroplast. I don't know if I reminded you of that, but this takes place also in the chloroplast, but this is in the stroma of the chloroplast, so outside the thylakoid. So if you go back all the way back to this picture of the chloroplast in topic 1, I just want to make sure we're all thinking the right thing here. The Calvin cycle is taking place outside the thylakoids in the chloroplast. So in this space out here, which is called the stroma, that's where the Calvin cycle is taking place. And so that G3P then, if I skip all the way back to the slide we were on, that G3P that we end up making, the plant is then going to use that G3P, like I said, to build lots of different things. That G3P can be used to build glucose. To build glucose, you need 2 G3Ps, and you can put them together to form glucose, a molecule of glucose, which is a 6 carbon molecule. But G3P is very versatile. Plants can use G3P and turn it into lots of different things as part of the plant's metabolism and all the different chemical reactions happening in plant cells. They can take that G3P and they can turn it into fatty acids and glycerol to start building lipids and phospholipids and cell membranes and fats and oils. They can turn it into other types of sugars. They can take that G3P and turn it into amino acids that can then be used to start building proteins, which do a million different things inside the plant cell. So this G3P will ultimately be used to build a huge variety of the organic molecules that we find in plants. But a lot of times that G3P does become glucose. And so to make a glucose molecule, C6H12O6, you're going to need 2 G3P molecules because each of those G3Ps is a 3 carbon molecule and glucose is a 6 carbon molecule. So you're going to need 2 of these guys. And so that's going to require the cycle to happen 6 times. So the cycle is going to have to occur 6 times to have 2 G3Ps that get to leave the cycle. Because remember, every 3 cycles, 1 G3P is made that gets to leave the cycle. So then you would have to have the cycle happen another 3 times, which will then produce a second G3P. And then you can make a glucose from that. So to make glucose, the cycle is going to have to happen 6 times, which is going to require 6 CO2 molecules, 1 per cycle, to make 1 glucose. You're going to need 6 CO2s to make 1 glucose. So that you can do the Calvin cycle 6 times. And then that's basically it for topic 3. I will point out one last misconception that students usually have. I just want to clarify real quick. A lot of times students have a misconception that photosynthesis is how plants make ATP for their cells. They think that, okay, in the chloroplast, there's photosynthesis happening, and that's what makes the ATP for plant cells. Because even in this picture here in the light-dependent reactions back in topic 2, I was telling you guys that some ATP is made, but that's definitely not the case. So in plant cells have a mitochondria, a different organelle, the mitochondria, that's in our cells as well. And the mitochondria, that's what his job is, to make the ATP for the plant cell. So in the mitochondria, we're making a buttload of ATP that's then used by the cell to power lots of different things happening inside the cell. Now in photosynthesis, there is some ATP that's being made during photosynthesis inside the chloroplast, But be careful because this ATP that's being made in photosynthesis, that ATP is immediately going to be used in the Calvin cycle. So we did make some ATP, but then we're going to use it immediately here in the Calvin cycle. And so really there's no leftover ATP during photosynthesis. There's not an abundance of ATP that's left over that the plant cell gets to use. Like this ATP that we're making in the chloroplast, that's going to stay in the chloroplast and then be used immediately during photosynthesis, because photosynthesis is going to require that those ATP molecules during the Calvin cycle, and there's none left over to share with the rest of the plant cell. So just know plant cells don't get their ATP from photosynthesis or from the chloroplast. They get their ATP from the mitochondria. That's where you have a huge abundance of ATP made for the plant cell to use. But in the chloroplast, we are making glucose, or we're making G3P, which is going to provide the building block to build all sorts of organic molecules, including sugar. That's the job of the chloroplast during photosynthesis. And then a lot of that will become sugar. A lot of that G3P will become glucose. And then plants can take that glucose that they built for themselves during photosynthesis, like right here. They can take that glucose and then send it to the mitochondria, and then the mitochondria can do cell respiration using that sugar to make ATP. But just know that that's two different things going on inside the plant cell. But anyway, that's it for topic 3. Thank you guys. I will see you later.